Shoot, Megan.
All right. Hi, y'all. Just holler if, someone holler at me if you can hear me or if I'm going. I usually have people flag me down when I'm going too fast. We're on Zoom now. So, today we're going to talk about everything but Varroa. And don't get your hopes up because I snuck a few Varroa slides in there. So you still get stuck with a little bit of Varroa. But we're going to look at pest predators, pathogens, and parasites. And so these will probably be like the 5 main sections. We're going to do colony assessments, what is killing our bees, enemies of the hive, diseases of the hive, and viruses. That's kind of 2 separate sections. And then resources. And a lot of the information that I pulled from this was from a presentation that I did with Dr. Natalie Steinhauer and the Bee Informed Partnership. And so I've updated it and modified it to fit current times and New Jersey specifically. And those are some of the resources for some of the different material, where it came from, a lot of the pictures. I don't have any of the information about where the pictures came from, but most of them came from Bee Informed Partnership. Not all of them, but most of them. And so in the top left hand of each slide, you'll be able to sort of get an idea of where that information came from. And so starting with colony assessment, before we dived into the diseases, I sort of wanted to go over some of the colony assessment stuff that I would look at when I'm doing a bee inspection, or what I'm looking for specifically when I start looking for said diseases. And so what do you see when you go into your beehive? Do you see worker bees? Are there nurse bees? Are there bees building comb? Honey stores, foragers? Do you see any drones? Is it that time of the year that you should be seeing drones? Do you happen to see the queen? Here you can see bees in all different phases of life doing their various jobs. And so the bee brood cycle, these are the different stages, just as a little refresher. So they're eggs for the first 3 days, and then they become larvae, then a pupa, and then day 21, they become an adult bee and they are born. They chew their way out of the cell. And so queens take 16 days and drones take 24 days. And so the progression of a worker bee that's female, the first about 16 days of her life, the time will be spent cleaning out dead bees or the little cells where bees have emerged, and they're cleaning out those cells. Then they'll spend some time being a feeder bee, where they'll help feed the young larvae royal jelly, and then they become, that's out of order. They're housekeeping bees in the beginning. I thought that was a little bit wrong. So they'll clean their own cell when they first come out, but they'll help clean out those cells. They'll work on stocking food for a phase of their life. And then towards the end of their life, they will develop glands that can build wax. So that'll be one of their jobs. They'll work on fanning to help control the temperature and humidity in the hive. They eventually will become a guard bee. That's when, that might be our least favorite stage of life. And then the very last stage of life would be foraging in their final 3 weeks of life. And so what I'm looking for, I'm going to look at the brood pattern when I'm going into a colony. We might look for the queen, we might not, depends on the situation. Definitely looking for eggs, larvae, catch brood. You want all stages of life that we just went over. You also want to see honey and some bee bread or pollen coming into the hive. And so healthy bees. Are your colonies building up really well? Do you have a solid brood pattern? Can you find newly emerged bees? Any eggs or white larvae? Or do you see a bad brood pattern or spotty brood pattern? Here you can see a picture of a spotty brood pattern. How did I do that? Sorry. So if it starts to look discolored or perforation, you should reach out to your state apiarist or me. Another great resource, obviously, is your beekeeping club. They should really be your first line for questions and stuff. There's so many people there that are able to help you. It's a really great resource. And then if the caps look normal, but you still have a spotty brood pattern, you might want to replace the queen. So signs of trouble. Here you guys might have seen this in the past. This would probably be the sign of a worker that her ovaries have kicked in and that she's now laying eggs. And so if the hive is um, queenless, this is going to happen. If they're queenless for an extended period of time, you'll have laying a laying worker develop. And so one solution with that is that you can try to requeen. Usually doesn't seem to work for me. Another option would be to, if you have a good nectar flow on and you have a strong colony, you can put newspaper between them and try to combine them after you've brushed some bees off of there to try to get rid of the ones that are laying. Here's another sign of trouble, a queen that's a drone layer. So if you start seeing a lot of drone cells that aren't consistent and are just filling in little holes, and maybe this works for you if you're trying to produce queens, but if not, then you might want to replace your queen. So this is another sign of a possibly failing queen. If you have unfertilized eggs in the worker cell, so here you can see it. Sometimes we call it popcorn. So you may want to requeen in that type of situation as well. And so another sign of trouble would be if we don't see eggs, if we only see capped brood, if there's emergency cells, or if the bees are agitated. And so one of your solutions might be to requeen the colony. But if there's queen cells like this, obviously you don't want to replace the queen. You just want to try to figure out what happened and why they're replacing it. Are they replacing the queen because they're out of space and they're going to swarm? So they're actually creating a new queen, or did the old queen fail, or, is she just gone maybe from the last inspection? And so, are there any questions for that section?
You look good so far. Landy's been answering questions in the chat, so don't see anything right now.
Okay. So, what is killing our bees? So, there's multiple interacting causes of death. And so, it's really important to remember that it's not just one thing that's going to absolutely kill your bees. I guess it theoretically could be one absolute thing, but usually with beekeeping, it's a combination of factors. And so it could be parasites and pathogens, pesticides, poor nutrition, habitat destruction, and monocultures. These are all things that our bees are constantly fighting on a daily basis. I mean, throwing a lack of rain or too much rain right now. But definitely a lack of rain could be a problem. And so one of the common questions that I get is colony collapse disorder, CCD. It was a big thing back in about 2008, but we haven't heard much about it. And we haven't seen a case of it in New Jersey really long time, like a decade. I think nationally they haven't really seen a case of it in a really long time. And that's because it is 6 very specific stages, or it's got like a specific checklist. So you have to have disappearance of most of the workforce, no dead bees in or around the colony, the presence of the queen, presence of capped brood, presence of food stores, and no robbing or delayed attack by pests. So we'd see no small hive beetle, no wax moth, and no robbing from other bees. And so those 6 things would be the only situation that you'd ever have colony collapse disorder if all 6 of those and nothing else was included. So some of the enemies of our hive, I'm going to start with some of the lesser enemies and work our way up to some of the ones that can be more of a pain. So ants and spiders. I think this is a really cool picture of a crab spider eating a honeybee. So a lot of times you'll find these in the feeders or near the top of the colony in the inner cover or outer cover. They're not a major threat for the bees. Fire ants can be a threat, but that's going to be more for us as humans. And then carpenter ants, of course, can chew up your woodware. So that could cost a little bit more. Or big red ants that come out of Florida. Sometimes they're fire ants, sometimes they're carpenter ants. The picture on the bottom left is actually red carpenter ants from Florida, last about 3 weeks ago in blueberries. So one thing that you can use for this is oil traps. I've never tried this, but That's what some people say works. So if you guys have any creative solutions for some of these, I'd love to see them in the chat later. Birds, so they're not a major threat. In flight, swallows and swifts will pick off bees. In the winter, woodpeckers can also be a problem on your woodenware. Robber flies could be a threat, but they're not a major one. I just love how the top right picture, he's mimicking a bumblebee, but eating a honeybee. So they're abundant in arid and sunny habitats. They're in the family of Scylidae. And those adults will actually eat wasps, bees, dragonflies, grasshoppers, and other flies. And so dragonflies, they're also not a major threat. They'll be they could be a major threat if you have a mating yard that's right near a bunch of dragonflies, because you'll have trouble getting your queens mated, because they'll pick off the bees right in the air. So they're also abundant in arid and sunny habitats, and they're usually near flowing water. Dragonflies usually like cleaner water, so that's why it should be flowing. Rivers, or if you have lake water that's moving through a dam or something, then you might have more dragonflies. So wasps and hornets. One sign for hornets or wasps is if you find headless bees at the entrance. It could be that they're getting picked off by yellow jackets or European hornets. And so if you have a weak or small colony, make sure you reduce that entrance. It doesn't matter the time of the year, but these will get really bad. For me in South Jersey, they'll get really bad in our dearth. So if you guys get like a late summer dearth, that's usually when they tend to get bad. So some of the other things that the bees will do is that if there actually is an intruder into their colony, like a wasp or a yellow jacket, and it's not a bunch of them, they'll ball up that intruder, start heating up their abdomens, and it cooks that intruder to death. So that's a really cool defense. Obviously, they can't do that if hundreds are attacking, but if a few come, they can sort of handle it. And then rodents. This is a good one. So they're not necessarily going to bother your bees, but the bees will propolis them if they're in the colony. They'll definitely destroy all your combs. So make sure you reduce entrances in the winter. I did not reduce one colony last year. I forgot to put my mouse guard on, and a mouse moved right on in. And I had boxes that looked just like this that they had destroyed all the comb in it, or about half the comb. And the problem with that is if the mouse is still in there, she'll start laying babies in the spring. And a lot of times the snakes will follow them in afterwards. So you got to be careful for that. So bears, that's definitely going to be a major problem. More in northern Jersey than South Jersey. But I'll defer to Landy on this one. So if you guys have any real bear questions, I think she's the go-to girl on this one. We don't have to use electric fences in South Jersey. There's been a few incidences over the past few years, but nothing major. And some other kind of pests for the hives can be raccoons, badgers, and skunks. And so pretty much this was everything exterior to the hive. Raccoons, badgers, and skunks. They're pretty not common that they'll bother hives, but you'll occasionally hear about them. And there's some different ways to fix that. But you can sort of tell if one of them is bothering your hive based on the demeanor of the colony. If they seem more agitated, if where you see scat, like this poop in front of the hive, you can actually see dead bees in the poop. Okay, so wax moth. Now we're looking at things inside the hive. So they can be larvae or adults. I consider them a secondary pest. They're moving in because the colony was already weak or there was too much space. They're moving in because there was an advantage that they could take advantage of. Something that was not perfect for them, perfect for the bees, so they took advantage of that. So some of the symptoms you'll see is that you'll have silk cocoons and or tunnels. So here you can see the different stages of the wax moth. You really want to keep the hive size relative to the population of bees. They can actually like burrow into this wood and make giant holes in it. And once you get rid of them, you can keep using that woodenware. And you can see the mothing that they made, or like the netting on that frame with all that pollen. And so for wax moth, frame storage is really important. They like dark areas, such as our colonies. So when you do have a wax problem or when you go to store frames, you want to make sure that they're possibly in the sun, but obviously you don't want the wax to melt. So you want it to be cool. And if there's airflow through there, ventilation is really important, or light. That's going to help deter the wax moths. Putting them in the freezer is another option, or paradichlorobenzene is a great option for storing frames. There's also another product out that's made out of BT. I haven't seen anyone using it yet in New Jersey, but I'm really curious if you guys have tried it. It's really important too to not use mothballs. The naphthalene is actually illegal in some places, and it'll definitely kill your bees. So don't use mothballs. And so wax moth versus small hive beetle. I get this question a lot. So one's a moth and one's a beetle. So their larvae are a little bit different. The top one's a wax moth. And what you can see here, he looks like a caterpillar. To be an insect, you have to have 6 legs. And so you can see the caterpillar at the top looks like he has a lot more than 6 legs. So he has 6 legs, 3 pairs right in the front on the right-hand side of the screen. And then the ones that you see towards the middle of the screen, the 4 extra sets of 2 there are called pro legs. So they're actually fake legs that just sort of help him wiggle along. And then the small hive beetle at the bottom, you can see he doesn't have any extra legs. He's just got that 3 sets of 2 there, right in the front. And their heads are a little bit different shaped, a little bit different color, and obviously they're different sizes, but at one life stage, they can be about the same size. And so small hive beetles, I hope you guys didn't eat dinner right before this, because some of these are pretty gross, but I do see this in the field sometimes, more than I'd like, actually. I'd say a couple times a year. So you can have the larva or the adults. This again is a secondary pest, so they're going to move in when something's not perfect for the bees, and they're going to take full advantage of something. So really healthy, strong hives that the space is proper for the population of bees. You shouldn't have a problem with that. So some of the symptoms would be a wet comb. You can see in the center picture how wet that honey capping looks, or maggot like larva, which obviously we can see here. So it's not much of a problem in the northern areas. Specifically, like Canada actually had a quarantine when small hive beetles first came to Canada, but they really do well in hot areas like Florida. And so they'll eat the brood and ferment the honey. And so here they are fermenting the honey. And you can see the adults at the bottom right and the larvae at the top center. So small hive beetle life cycle, I think this is really important to understand, just because then you can try to figure out how to control them. Everyone's going to, not everyone, a lot of people are going to have them in their colonies. If you're seeing 5, 6, 7, that's not crazy. Even if you're seeing, 10 or 20 running around a really large colony, I think that's still really manageable, and the bees are able to handle that. It's when you start getting into the hundreds and thousands that it becomes a problem for the bees. And so the eggs are laid in the hive. The beetle lays eggs in the hive. The larva of the beetle will then eat bee brood and honey. They will then leave the hive and dig into the soil for metamorphosis. So if you have screen bottom boards, it's really easy for them to drop straight through to the bottom down to the soil. If you have solid bottom, obviously that larva is going to have to find another way out. So the adult will emerge from the ground and climb back into the hive to go find a mate. So here's some good pictures of small hive beetles. And one really neat thing about small hive beetles is their feet. See on their feet. So their toes are shaped like a fishing hook. And with a fishing hook, it can get trapped in things. And so one of my favorite control methods for small hive beetles is actually to use unscented dry floor cloths or Swiffer cloths, because their feet will get stuck in it. But the honeybees don't have that fishing hook shaped toe, so they won't get stuck in the cloth as much, at least. So main ways of small hive beetle control is really through traps, or obstacles between them and the soil. So plastic tarps, drenching the soil with chemicals. Chickens are really good at controlling them. But here's some of the different traps that are out there on the market right now. And so there's different small hive beetle chemicals that you can use. 2 of them specifically. One's for the soil. A really toxic chemical, so be really careful about how you use that. I don't know many people that have used it. And then in the hive, there's another product called Checkmate Plus. It's also a varroa mite control, but I would highly suggest not using it ever, 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 because it's shown to be resistant. Bees have been shown to be resistant to it, and the product subsides and it lasts for a really long time. So you really want to avoid that product. That's not really a viable option in my opinion. And then there's also soil nematodes that you can use. And so here's a picture of a really zoomed in picture of a nematode. But they'll actually live in the soil and feed on the pupa as they're pupating in the soil. So, any questions on those?
So, Megan, up here, I'm curious if anybody listening to this talk has ever had more than like 20 hive beetles. It's really interesting to see the various controls, but they don't seem to be an issue for us, at least for me, except in the honey house. And there, it just means I need to extract my crop within a week. That's like the outside limit.
Yeah, I was gonna say usually it's about 3 days you want to extract your honey supers.
Yeah, that's ideal. After a week, I might start to see a few hive beetles crawling around, but I'm usually still okay, unless the weather is super, super hot. But yeah, so you would say 3 days?
Yes, about that.
We've got a question from Bonnie. She couldn't see where you put the Swiffer sheets. Was it on the bottom board under the screen, or?
That's a great question.
Yeah.
So what I do with the Swiffer sheets is I cut one in half, and then I put it at the corner between 2 brood boxes, or between a brood box and a honey super. I did not include a photo of that, but I'll definitely update the talk to include that. But I can get you guys a picture of that.
Okay, great. Thank you. And I think everybody else is good. I don't see any other, let's see. Oh, Adriana said she made the mistake of buying scented sheets.
Yes.
What happens if you buy scented sheets?
I don't know the answer to that. Maybe Adriana will tell us in chat for the next little mini break. All right, so I'll keep going, and Adriana can update us at the next mini break. So diseases of the hive, I'll stop a few times through this. It's a really long section. So this is mainly pathogens and parasites. It's going to be everything in the colony, the really itty-bitty, nitty-gritty stuff. So nosema. I think you could just put a giant question mark on this slide, and that would answer everyone's questions, because there's still so many questions around nosema, specifically in research and science, and what this actually means for a colony. Is it a true health marker? Is it something we just have to deal with? What does it really mean for the colony? So nosema, there's 2 types of nosema: Nosema apis and Nosema ceranae. It's a microsporidian unicellular parasite. It lives in the bee's intestine. Nosema apis can be called wet nosema sometimes, and ceranae sometimes is called dry nosema. So the symptoms will be dysentery, or distended abdomens. I don't know that I've ever seen the distended abdomens, but I think seeing that dysentery on the bee boxes is what most people think of right away when you think of nosema. And so Nosema apis is usually associated with spring and fall disease. Wet climate, when cleansing flights aren't available in the winter, that's when you can have problems. You really want to make sure the location of your hives is not too humid. And then Nosema ceranae is usually associated with summertime, and weak colonies. If they're not foraging, that can be one of the problems. And so prevention for nosema is to make sure that there's really healthy, rich pollen coming into the hive, or pollen producing plants around. And so I know that's really hard to think about, but when you're out doing a bee inspection, look for pollen, see if it's coming in, look for multiple colors to see if they're going to different resources. If we had a really long spell where the bees couldn't get out, maybe feed a little bit of pollen substitute to them so that they did have new pollen coming in. There is medication that can be used for Nosema. It was originally developed for Nosema apis. So it's been redeveloped again. So, but still not really sure how well it works and how effective it is, especially for that second type of Nosema that we started showing up. I'm not sure when it started showing up, but it's pretty much taken over. So we really see mostly Nosema ceranae, I believe. And so there's transmission can be spread through the spores in the fecal matter or through oral contact. And so Nosema diagnosis, those are little Nosema cells in the top right corner, all those little neon highlighter shade sesame seeds. And so you can use a hemocytometer to count. There's a couple of beekeepers in New Jersey who do this for themselves. The Department of Ag does it too if you need it done. And the USDA also does sampling for this. However, not right now during COVID. They're still trying to catch up. So there are some options to get some testing if you want it. But pretty much you take a sample of bees, squish them up with your best rolling pin, mix them with some water. You have to do a specific amount of water for the amount of bees in there. And then you'll look at it on the hemocytometer and count specific cells in that grid. And you'll be able to get a number of how many Nosema are in your colony. And so Nosema control, it's one type. It's called fumagillin. I'd say the jury's still out. The original formulation was effective in controlling Nosema apis infections. Its significant biological activity against Nosema ceranae was shown in 2016. And then there's a higher risk of bee mortality in cage studies also in 2016. And so this is the second time that this product was developed. And the product is fed through sugar syrup during the spring and fall to keep the spore counts low. So if your bees do have Nosema, again, it doesn't mean your bees will die. It's just trying to figure out what this really means for honeybees, especially, we use it for a lot of health markers in science and research, and it makes you wonder if it really has mattered, just because I've seen colonies with really high Nosema loads, and they are perfectly fine, healthy bees otherwise. And so if you do have Nosema or equipment that has a lot of the diarrhea on it, you want to decontaminate that by freezing it or placing it in direct sunlight for 72 hours. So tracheal mites, this might be something you guys haven't heard of. Maybe some of you have. It's in the adult bee trachea. They're really, 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 really little, microscopic. So some of the symptoms, there's no one telltale sign of this disease, but something that can be seen is to have K-shaped wings. So that's something I'll often look for in my inspections. Here you can see a bee with a K-shaped wing. You can also see deformed wing virus on the bee above it. But bees often crawling on the bottom board or appearing morbid can be that. And so that's actually the trachea of a honeybee with tracheal mites in it. And so it has not been a major problem since the early 90s. One of the preventions is to have resistant stocks such as buckfast or Russians. It's also really not a problem anymore. So as long as your bees are acting healthy, it's not something to be concerned of. And we'll get to that in the next slide, why. But methods of detection, microscopic observation of the trachea and the breathing tubes, and an inspector can do sampling for that. So if the percentage of infested adults is 10% or greater, treatment is warranted. You would want to treat the colonies in late summer, autumn. And one of the reasons it's not a major problem is because when we started getting varroa mites in the 80s, a bunch of different products were developed for treating varroa mites. Well, some of those, specifically the thymol products, were also effective against tracheal mites. So if you're using Apiguard, it's also going to be effective against tracheal mites. So that's one of the main reasons we don't see it anymore. It's just that a lot of varroicides are also effective against the tracheal mites. So here's another one. This is a really fun one. Chalk brood and stem brood. So what you're going to see here is chalky looking larva, almost like a fried egg, or a mummified little body. You'll see the mummies at the bottom, on your bottom board or on your screen bottom board or right at the entrance of the colony. So it's a fungus. It'll be in the open and sealed brood. It usually arises in the spring, specifically when we have really wet springs. You want to check your hive ventilation and pollen stores. It's associated with stress. Our bees can handle a couple, different stressors throughout the bee season or throughout the year, but they can't handle a lot at once. And so this is related with stress. And it's not too much of a problem. It usually cleans up on its own if you get good pollen in there and plenty of ventilation. So it's a visual detection, and there's no chemical control for this. Requeening can be beneficial if your bees are more hygienic. This one, to me, on the left looks like a little fried egg, sort of on its side, but still a fried egg. So it's something to look for if you are looking at bees in the spring. And then, so these are some of the pathogens. We talked about chalk brood, and we're going to continue on to American foul brood, European foul brood, and sac brood. So American foul brood, I think this is a really great picture. A lot of the really good pictures were taken by Rob Schneider from the
Meghan?
Informed Partnership. Yep.
Can we ask a couple of questions before you move on?
Yeah.
Okay, great. Because I've got a couple of good questions. Adriana wants to know if you need a veterinarian to prescribe fumagillin.
No.
And I didn't know the answer.
I could buy it on Man Lake's website today, so I do not think I needed one.
So I guess you don't. Let's see, what else was there? Symptoms, overt symptoms of nosema. Okay, Gail's asking, how do you diagnose? She said Apis cerana, but she means nosema. I think she means nosema cerani. And I'm one of those people that, with the aphis study, I was part of the aphis study for many years.
Yep.
Always had nosema in
Yep.
the samples, never had any symptoms.
Yep.
So, like over the years, I mean, back when we got it, when we got nosema cerani, it seemed like the first couple of years there were colonies that were symptomatic, and the symptoms were that they just kind of sat there. There was nothing overtly wrong in the brood nest. The brood pattern looked okay, although small, but the bees never grew and they didn't eat. So they wouldn't take syrup the way a normal colony would. And those were the only symptoms that you could see at a colony level without a microscope.
Mm-hmm.
But, I haven't seen that for years and years and years. And every time my bees get tested, they have nosema. So have many of them become resistant? Just like tracheal mites are pretty much a thing of the past? What do you think?
That's a loaded question. I think that, it's a great question. And I think going back to just, we really don't know. When I was at University of Maryland working with Bee Informed Partnership and doing a lot of the Varroa nosema testing, we were using nosema as a marker for health. But I can tell you personally, like Landy, that through the aphis study, when my bees were sampled in Delaware, my Delaware bees were sampled, they came back with the highest load of nosema in the state. And I was like, excuse me, you did something wrong. Because my bees were healthy. I was getting 0% loss in the winter, or I might lose one colony out of 20. So to say that I had the highest nosema in the state and it was really high, and I, something to be concerned of. I think it's really one of those big question marks. It's definitely something to consider if you are seeing that dysentery, that getting healthy food in there for them. Hopefully they're taking that healthy food, that healthy pollen. But I really still think it's a major question in beekeeping that researchers just overlook, because I guess it has kind of gone towards the past. We're on to new and exciting new viruses, new pests and parasites constantly. So I do
How
think it's
wonderful.
something that's over. Yeah,
And
so
the only other thing I wanted to ask about nosema was, my understanding was that dysentery is not a symptom, or at least not a reliable symptom of Serrani. Um, it wasn't even really that reliable for Apis, although you did see it fairly frequently with Apis. You would get dysentery, but I thought that you can have Serrani and no dysentery at all. Um, have you heard that?
yeah, I mean, my bees didn't have any dysentery, and
Yeah.
they were,
Okay.
millions and millions of spores per bee, and that's really
Yeah.
high.
Yeah. Me too.
So
High
yeah,
levels. Perfect
it's
bees.
and to test for Nosema ceranae versus Nosema apis, they have to do it in a lab, obviously, but it's not something they even really do that often at the USDA,
Mhm.
because they were getting that it was like 99% Nosema ceranae,
Right.
and hardly ever finding apis.
Okay. All right. We'll let you move on now.
Cool. So we're getting to the juicy stuff. So American foulbrood, it's also called AFB. It's the Pentabacillus larva. It's a bacteria. It will affect the open brood. That's something really, really important to remember for EFB and AFB is the different stages of when the larva is affected and when the brood is affected. So both of them are going to affect open brood, but you'll see later how it kind of affects them down the long run. So you'll have larva putrefaction, black scale and odor, really bad odor. What It's hard to describe the odor, but it's something that once you smell it once, you'll never forget it, if that makes sense. And so it's going to be in the sealed brood. We'll have a discolored or sunken capping, or punctured capping. And then you can also do the rope test. And so in this picture, we can see some dead pupa right in the center. They're brown, they're sunken, they're gooey, they're just looking slimy. I know it's hard to see, but you can see the scale in a couple of these cells. It's going to be in the bottom. And then you can also see some of the sunken and punctured cappings. But we also still have some what look healthy white larvae here. And the reason that those two brown larvae are showing, it's probably because the bees uncapped them, or the researcher uncapped them for this photo. Because American foulbrood is going to affect the larva once it's capped. So it's spread through the food. So the bees will feed the food to the baby bees, and then they'll cap up that cell, and then it'll take effect while it's got that capping. And that's why you end up with these sunken, discolored, or perforated cappings. And so, again, the top picture, you can sort of see a little bit of scale. It's going to be the black shiny stuff, just south of where that bee is. You can see a couple shiny cells. And so prevention is really important for American foulbrood. It's really the only option, I think. It's not the only option, but it should be your main option. You want to have good beekeeping practices. So I know Landy covered this definitely in the beekeeping class. Make sure you're being really clean about if you're going to another bee yard, using that bee yard's equipment, making sure you always have a clean hive tool, gloves. That's really important when I'm going around inspecting for the state, to make sure I have clean gloves, or clean hive tool, clean smoker. So having hygienic stock of bees, avoid robbing by keeping colonies strong or minimizing entrances. You want to minimize comb swapping between hives. If you know your operation and you really know what you're looking for, that's great. But if you just want to go to, Bob down the street because he has some extra stuff for sale, you really want to be careful that you're not picking up any American foulbrood. And so different places have suggested replacing 3 combs a year in your brood comb. So that bottom picture, you can see how black that comb is. I know Tim Schuler and a lot of other respected beekeepers say if the bees are still using it, it's still fine. I probably fall in the camp somewhere between there. As a growing beekeeper, I'm always running out of comb, so it's hard to afford getting rid of comb when I always need comb. But at the same time, you want to be really cautious about receiving old comb from someone, especially if it's dark like this. And for the price of frames, it's not really worth it. So, and so since 2017, beekeepers are not um, to do preventative treatment or prophylactic treatment with antibiotics, and that's due to the veterinary feed directive. Um, and so American foul brood is really contagious because it produces these spores that can spread. They're pretty resistant over time, fire, freezing. So you just got to be really careful. And so here's some of the different testing that you can do for detection. If you think you have it, call your inspector, me, for a sample, or reach out to someone in the bee club. They're definitely, Landy's going to know what it looks like, and a couple other, I'm sure a lot of other people in the group will know what it looks like. And if they really think it's American foul brood, they'll call me in right away. And so you can do this rope test. And so you would stick a stick, cotton swab, something in there, corner of a hive tool. And if that larva is going to rope out like that, then it's probably American foul brood. A lot of the other diseases will look snotty like this, like snotty brood, but they're not going to rope out. And so there's a Vita test kit that they do. It's pictured on the right. USDA and Department of Ag here do testing. And then there's the Holtz milk test. I haven't actually used this, but it's always another option. So you can see a picture there of the different testing. So American foul brood, the treatment is to destroy your hives with fire, or you can double bag them and I'll remove them safely to a facility that will incinerate them. The other is to use antibiotics. I really don't think this is a good option. You would treat the colonies 8 weeks prior to the nectar flow to prevent honey from being contaminated. And for this, you would definitely need a veterinary feed directive or a prescription from a veterinarian. And the 3 products that can be used for American foul brood are terramycin, Tylan, and lincomycin. Terramycin is the most common. This stuff is just going to persist in your colonies, though. So you really just want to get it out if you have it so that you can keep everything else clean. And so this is something I think is really exciting. We're talking about all these negative things. So I had to make something exciting in the talk. And so kind of the future of American foul brood is these bacteriophages. And so it's going to become a product that will eventually be on the market. It's an alternative or replacement to the conventional antibiotic treatment. And so you wouldn't need a veterinary feed drive or prescription. It's still in testing. They've done a lot of research in Utah. It's got a really powerful potential. There's a couple of private companies and universities doing research. And so that's an actual picture of a bacteriophage attaching to foul brood. I don't know a lot about it, but here's kind of a graph if you're interested in how they work. You can Google it too, but Utah has shown a huge decrease in American foul brood. They were probably the second highest in the country for cases of foul brood, and they've really decreased. So it's looking good. Back to the negative stuff. So European foul brood. So again, it's a bacteria. This one they'll often refer to European foul brood and friends. There's a lot of other diseases that look like European foul brood, and then there's also a lot of other things that kind of get in there and make it all gross at the same time. And so the symptoms, the brood will be a dull white, and it'll eventually become a yellow, almost brown to eventually black. The age of the dead brood is usually younger. It's going to be the unsealed larva. And so that's the big difference. AFB, it was in the sealed larva. European foul brood, it's going to be before it's capped. And so the consistency will be rubbery or granular. It's not going to be elastic. So the prevention for EFB, it's mainly caused by stress. So maintaining a strong, healthy colony is the best prevention for this disease. If we have a not stellar spring where the bees are really stressed for resources, providing those resources can help keep European foul brood out of your colony. And so the detection is visual through a test kit, or you can get samples done for USDA or New Jersey Department of Ag. And so the treatment, you want to verify the infestation and distinguish it from other brood diseases. So here in the picture, you can see some healthy brood and you can see some yellowing larvae. that are sitting in that really yellow, nasty brood food or liquid. Some of the larvae you can see there are yellow in the bottom center. And then one thing that I look for as a symptom that might be pretty hard to see, but in the white larva, you can see the tracheal system. So those really exaggerated white lines, that's going to be the tracheal system of the bees, that how they breathe. And that's another sign for European foulbrood. And so with colonies that have a light infection, you want to reduce the brood nest area. You can replace any of the infected combs if it's really, really heavy infected on a specific comb. You want to really keep that population strong. Another option is to use terramycin, but then again, you need a veterinary feed directive or a prescription. And for all cases, you really want to keep that colony stuffed to itself. So if you know you have EFB, inspect that colony last, and then go inside, wash your hands, clean your hive tool, stuff like that before you would move on to another colony. Any questions on AFB or EFB?
Yes, there were a couple of questions. I tried to answer them in the chat, but can you talk about one person asked a question about burning frames versus burning the whole hive.
Yeah,
Can
so
you-
they say that you can just pull out the frames. You're supposed to dig a hole in the ground. And again, a lot of this stuff's going to pop up probably in the summer, where you have to be careful about burn bans in the state, or starting forest fires. So that's another thing to be really cautious of. But you would dig a hole in the ground. You would put in those frames and then burn them, bury them. And another option, what you would then do with the boxes, would be to scorch them. Again, I don't want this stuff getting into my operation because it can persist for hundreds of years. So, you could have acquired your great grandfather's equipment when he moved here from Wisconsin and he was a beekeeper, and that stuff could still be living in those. So for me, the best solution that I've found for me handling it, because again, if you have plastic foundation, it's going to be really hard to burn that, or close to illegal to burn plastic foundation. I just triple bag it, and through the state, I have a permit that I'm able to haul those away to an incinerator and get the plastic burned. So it's also, might not be the most ethical way to kill the bees in there, but if you triple bag them or double bag them with really nice, heavy contractor bags, those bees aren't getting out. And you're going to contain that situation right there.
So the other question is, can you just touch briefly on shook swarming adult bees during a nectar flow? That's at your option as state apiarist. Can't be done during dearth, but that can be done, right, to save the adults.
Yeah, so a shook swarm, for anyone who doesn't know, would be essentially taking the whole colony and shaking it onto brand new foundation, brand new boxes, brand new equipment. I still think you would be treating with one of those antibiotics in that situation.
Yes.
But the idea behind that is because any of the infected food that the bees have in their gut, since you're putting them on foundation, they're going to have to use that food to build wax. And so that food's going to get processed through their body, and that actually somehow magically removes the foul brood. So I've actually done that with really, really bad cases of European foul brood. So it's definitely an option.
Okay.
But you would definitely want to treat with it too.
Yeah.
So.
And you could never, you would never want to do that, for instance, in August or, during dearth. It's got to be during a nectar flow, from what I understand, to prevent
correct.
robbing. Yeah.
Yeah. Prevent robbing. I mean, a lot of this, you got to think about even the cost too as a beekeeper on the resources that you have into that colony.
Okay, great. You were breaking up there for a minute, but I think we're good now. Adriana's also asking, can we get a vet associated with the club? I thought I needed one a few years ago and it was expensive, and you have to pay for the time of the vet to come diagnose the hive. It's a good point. It's still an issue.
That's a great question. It's definitely still an issue. I got a text today about it from another researcher in New Jersey. So it's on my list to look into. It might just be a winter project. If that makes
Okay.
any sense. So.
Okay. Thanks, Megan.
Cool.
Move on.
So moving on to viruses. Hopefully you guys are still awake, and I can't see you if you aren't, so that's okay. So viruses. They're microscopic organisms. It can be in the RNA or DNA. They do not require their own nutrients to live independently. Only multiply in a living host, and so that living host can be a bee or a varroa mite. And then once the host cell becomes damaged or dies, it releases a large amount of infected virus particles. So viruses and honeybees, they've been known to scientists for 50 years. They were pretty much harmless until the 1980s when the varroa mite became widespread because it could transfer them. It became a vector for them. There's about 26 or so viruses have been discovered. The majority of them are associated with the varroa mites physically, or they could be a biological vector. Controlling varroa populations in a hive will often control the viruses. That's a tricky one, though. You really want to make sure you control your varroa population before it gets too high. Finding symptoms of the viral disease is indicative of a varroa epidemic in the colony. There's still a lot of emerging information coming out. Viruses are still something we really don't understand totally. They typically infect the larva or pupal stages. So that's when they'll be infected, but the symptoms won't be obvious until they're an adult. Viruses can be consumed in pollen or jelly produced by the nurse bees, transmitted by varroa. Viruses can remain in a latent form within the host, acting as a reservoir of infection, complicating diagnosis and control, and only becoming an outbreak when conditions are right. And so this goes back to having stressors. And so if your bees have a lot of stressors, that's also a perfect time for a virus to present itself and not be just hiding in the darkness. So detection of viruses, it's really difficult due to the size of them. It's really expensive to get it done. Overt infections can be visible, but there's also a lack of symptoms. Doesn't rule out the presence of this virus. You guys will see in the next couple slides. I think it's pretty shocking. So this is research that Landy had talked about already from the National Honeybee Survey done by APHIS. And so some of this data is a little bit older, but the trends are still going on. They're a little bit behind in getting information out, especially with the pandemic going on and just, getting that all done. And so here we can see two viruses that I circled in red, deformed wing virus on the right and black queen cell virus. And so black queen cell virus from 2009 to 2016, they found that at its peak in almost 90% of the colonies that were tested. Deformed wing virus probably had an average of about 80% of the colonies. So think about all the beekeepers in Jersey or even your bees, and if it's in 80% of the colonies, but yet it's just not presenting itself. It's laying there latent until they get a stressor. Maybe that stressor is European foulbrood, something like that. So I think that's pretty amazing that there's two viruses that can be so prevalent, but we really don't see them because they're laying latent. And so here's some of the most common viruses. You have acute bee paralysis virus, black queen cell virus, which you can see on the top right, deformed wing virus, chronic bee paralysis virus, Israeli acute paralysis virus, cashmere bee virus, and sac brood virus. And so sac brood virus is at the bottom right. And a lot of things that you'll see with these viruses is that one thing in common is a lot of them say paralysis. And so many of them prevent the bees from moving around the colony. So sac brood, the symptoms are that you're going to have dead larvae that appear watery and granular with a thick skin that forms as a sac. So that's kind of the later stage of the sac brood. The first stage is that you'll have a small head lifted towards the top of the cell, resembling the shape of a canoe. I describe it as a little alien pinhead on top of a larva or pupa that's trying to do a sit-up in its cell. And so here you can see a little bit of the different stages. There will be a quiz on this later. My favorite bee disease, sac brood. You guys got to remember that. So prevention, it's caused by stress. So again, we talked about that stress. So maintaining a strong and healthy colony is the best prevention for this disease. Here you can see that sac that someone pulled out with a pair of tweezers. And you can even see on the bottom picture how healthy a lot of that other brood looks, minus those few cells that have the sac brood. We see this in New Jersey a little bit. We don't see it a lot. So there's no treatment for it. Requeening may be helpful if it's more hygienic, but maintaining a strong colony is your best option. A lot of times I'll see sac brood in association with a colony that has European foul brood or chalk brood, because a lot of these stress diseases, they're going to move in when it's advantage for them. So deformed wing virus, this is probably the most common. It's widely distributed. It's found in all castes and life stages. It'll persist in adults without obvious symptoms. It's associated with the varroa, directly linked to varroa infestations. It's also transmitted through food, feces, and queen to egg, and from drone to queen. And so it can affect aggression and learning behavior of the adult bees as well. And so deformed wing virus can actually replicate in varroa. This was the first virus for bees that was shown that it didn't need to replicate in just honeybees, that it could also replicate in varroa. It's strongly associated with winter colony mortality. The controlled deformed wing is usually achieved by treatment against varroa. This is where all my varroa slips in here. The infection should be reduced in a colony far in advance of winter. We want to make sure our bees are healthy going into winter. It's closely related to a couple of the other viruses. They're kind of all in this deformed wing virus complex. And an acute infection where you have a lot of bees, you're going to have early death of pupa, deformed wings, shortened abdomens. Shortened abdomens are also a symptom from varroa mites. Just a little tidbit. And cuticle discoloration in adult bees, which will die in 3 days, causing the colony to eventually collapse. Sorry. So something here from, I'm gonna grab water real quick, sorry. So here's something from the honeybee survey again, which I thought was really interesting. So if you look at each, for previous years of the survey from 2010 through 2018, you can see that the lowest levels where they found prevalence for deformed wing virus was 65%. So deformed wing virus is out there. I definitely don't see it in 65% of the colonies I go in, but it's definitely laying in there latent, and they can detect it at levels a lot lower than when that virus is actually going to present itself for us to know that it's there. So varroa destructor virus is another one. This is probably one of the more new viruses. It's also called VDV. It's an RNA virus. It's closely related to deformed wing virus. It's also been called deformed wing virus strand B. Just discovered 2016, 2017. They're not really sure, and it's associated with varroa. And so here's a map that was Bee Informed Partnership and the National Honeybee Survey. They were able to go back and look at historic samples in 2017 from 2010, which you can see on the left. And from those states that they tested in blue, they found that was in Indiana and Pennsylvania back in 2010, and we didn't even know about it till 2017. And so just in 6 years, you can see how much that virus spread around the country. So I also think this graph is just shocking when we're looking at prevalence of a virus that we didn't even know existed, and how quickly it spread throughout the United States. Again, here, you're looking at peaks in the fall and coming out of winter, which makes sense with a varroa associated virus. And it's just, there's months where it's 100%. There's 3 months. So it's definitely very prevalent. We don't really know what it does besides that it's similar to deformed wing virus. So another one is chronic bee paralysis virus. I think I get a lot of questions about this, but there's not much that you can do. It's partially open and dislocated wings, so they could be shiny bees, black bees, dark bees, not just typical black bees that, your bees are all black, but they'll look really hairless and greasy. Um, the infected bees are picked out of the colony. It sort of looks like a robbing, but they're actually being removed. Sometimes they'll be trembling out in front of the colony, but you'll see that K-wing a lot too with these. Um, so it's not correlated with varroa or nosema. The prevalence is on the rise throughout the country, but there's no known treatments. The one way to prevent spread is to quarantine any colonies that have this. You can requeen to try to have more hygienic bees, frame exchange between colonies, and limiting barrel feeding or open feeding, just because the bees then have a lot of chance to come in contact with each other. So here's another virus. Here's a really good example of how we just really don't know anything. Lake cyanide virus 2. So there was a 1. It's related to chronic bee paralysis virus. It hasn't been described yet. They've genotyped the virus to figure out some of its heritage, but that's really about it. It's highly abundant in weak colonies and healthy colonies. So what's it really mean? We don't know. It's just one of those viruses that's out there. But here from the National Survey, you can see that they've been testing for it since 2010. And I mean, that prevalence peaks about 70%. So a virus that we haven't even described, have no idea what it does, but it's in 70% of the colony. So, it shows that there's a lot more research to do in beekeeping, specifically with viruses and diseases and what it really means for us as beekeepers. And so this is one of my favorite analogies called the doorknob effect. So honeybee viruses are not limited to just honeybees. They can be found in other non-apis bee species. Small hive beetles, pollen, and nectar has also shown to have them. So non-infected individuals or colonies can occur during foraging on common flowers. So it's just like going up and touching a dirty doorknob during a pandemic. You leave virus particles behind, or you don't, but you might pick up someone else's virus particles. And so these two bees, it's been shown with this with deformed wing virus that it can be spread on a flower from a honeybee to a bumblebee. So by us being beekeepers and wanting to help honeybees, and I mean we should be good stewards for all bees in general, the native bees, our honeybees. We really need to make sure we manage our varroa mites, so that we don't inadvertently let one of our bees infect a bumblebee. So here are the varroa slides. So Honeybee Health Coalition in the top left. It's a really good resource for varroa information. It's our number one enemy. You have to have a management plan in place. This is definitely the time of the year. If you haven't decided how you're going to manage your mites this year, that you need to make a decision soon. So we're right at that peak population for our honeybees right now. We might still be increasing in a little bit of spots. Some places might be decreasing depending on weather, foraging, stuff like that. But you can see there that the orange line is our varroa mites, and those are still increasing. Even when our bees start to decrease in the fall, those varroa mites will still be increasing. And so varroa, definitely want to have 2 types of treatment per year before the honey flow and before winter. And you want to treat all swarms, especially the later in the season it gets. A lot of times in the fall, those bees are absconding and getting away from a disease instead of just swarming due to space. It's really important to monitor and check before and after for effectiveness. And so this is a little bit older. It's from 2015, but it came out of the USDA lab in Beltsville, Maryland. I think it's a really neat image to really show how effective some of the treatments are. Cumulus, which is check mite plus, the first thing on the left. Apistan, the second one. Apar is the third. And then the fourth one all the way on the right is Flumet. It's a product in Canada. It's not labeled for use in the United States. And they tested United States bees. You can see the text on the right from the survey for the national honeybee. And they were testing those varroa mites to see if they were resistant to different chemicals. And you can see here where they were resistant to coumaphos, slightly resistant to apistan, which is fluvalinate, and flumetharin, the Canadian product, which we've never even used in the United States. Theoretically, we've never used. It's never been labeled for use. But the apivar was still pretty effective back in 2015. So I'd really like them to do this research again. Once we're past this pandemic, I think the USDA lab will really get up and swinging, and hopefully they'll redo the study with some of the newer products that are out on the market as well. And so this is why it's really important to do varroa monitoring. The blue boxes are average mites per month. So you can see we're end of June, well, beginning of June. Just lucky I know what month it is. So yeah, so that is there are 2 mites per 100 is the national average for June. So you want to kind of think about that if you're doing mite sampling. But you can see here that red line, the beekeeper, was above that threshold of what is considered 3 mites per 100. And so they did a treatment in September, which is pretty late to be doing a treatment. It's probably too late, but it was a treatment and it was effective because he tested again in October and you could see that he had a lower number. However, this beekeeper, you can see his mites are way higher. He did a treatment in September or August. You can see that the mites drop in September, but then all of a sudden they spike again in October. And so it wasn't an ineffective treatment because obviously those mites dropped for a month. So was it some type of reinfestation? So this is some of the work I did at University of Maryland and why it's so important to monitor varroa. The red circle in the center, we crashed 5 colonies due to varroa mites. And so these bees in the varroa, they're not just going to sit there and die with the ship. They're not the captains. They're going to look to get out and go somewhere. And so if you bring food, your neighbors are usually pretty welcoming. So all those red lines, it's about a half mile to about 2 miles, I think. We found bees from those crashing colonies, which were painted bright yellow, in healthy colonies. So I don't think it says on there what it was, but the varroa mites were really high, like 15 per 100. And so if you had a really high mite count, you would think for every 100 bees that went and visited or moved into a new colony to find a new home, 15 of them would have taken a mite with them. And so that's how you have a lot of the reinfestation. With that project, I also learned don't paint bees yellow, pollen's yellow. It's really hard to see sometimes. So you really need to monitor for varroa. Living up to its scientific name, it causes premature aging of adults. It's a less effective worker force, death of developing pupal brood, feeding on host. It results in the transmission of, and in some instances, pathogenic viruses and other diseases. And it can reach epidemic portions and kill the entire colony, or they can abscond. So here's my contact information if you guys have any questions. You can email me. That's probably the best way, or texting, honestly. There's my office and cell. This is my favorite tool for varroa management. It's through the Honeybee Health Coalition. They have a great manual that walks you step by step through each product. It talks about different times of the season. There's even a sheet in there that you can use for recording data out in your beehives. That's probably the number one question I get asked when I go to a bee inspection is what's the best way to record data or manage all this information I have about my bees. And everyone's different, so it's going to be different. But if you need a starting spot, this is a great place to start. And then they also have this decision tool that's really useful so that if you said, hey, it's, June right now, we're at the peak of our season, I still have honey supers on, you can put all that information into the decision tool. It'll say, do you want to make the decision to use a synthetic product? Do you want to use a natural product? Things like that. And it'll help you come out with the options for what you want to do for your beekeeping. And then here's the testing that the New Jersey Department of Ag does. They test for nosema and varroa, and then European foul brood and American foul brood. And you can mail the samples directly to our office. They do it downstairs in the lab. My office is on the second floor. But yeah, if you have any questions about that, feel free to reach out. That's it.
Thank you, Megan. That was an excellent talk. Even with the Varroa at the end, you couldn't resist.
Never. It's my favorite.
Yeah, well, it's hard to resist because it's so important. We did have, let's see, we have a question from Adriana who says, if we get a June swarm, do we forego honey production and treat with Apivar? I thought she could treat with Formic Pro and still get her honey, but do you want to comment on that?
Yeah, I think one of my favorite questions for beekeepers is you got to think, or, when you're thinking about your bees, what's your goal in your apiary? So if your goal in your apiary is to produce a lot of honey, maybe you have to figure that out exactly for how you're going to produce the most honey. If your goal in your apiary is to produce a lot of wax because you need draw and comb because you ran out of frames in February or March, me, for example, and you've been catching up. So you have to think about what some of those goals are for your apiary, or what, what you're trying to achieve in your area, or is it, a business for you and you need to make certain things happen, or management decisions. And so I think that really varies based on what your goal is in your apiary. So I didn't answer it.
Megan, I'm going to post a link in the chat for everyone directly to the Honeybee Health Coalition Varroa Management Decision Tool. That is a really cool online tool.
I love it. It takes the thinking out of it almost.
Yeah,
Almost.
I mean,
Yeah,
this is really nice.
Yeah, it's excellent. Megan, I've got a question for you. So you might even be too young for this, but 20 years ago, 25 years ago, when I started keeping bees, we put on Apistan, right, fluvalinate, once a year. You treat it in July or August. And as long as you got a treatment on by the end of August, you were fine. Your bees would live right through the winter. It was no big deal. But I'm wondering, and obviously we can't do that now. Treating and testing, I think that the minimum successful treatment protocol is probably about three treatments per year. And I've had a couple of, I've done 4 and even 5, depending on the test results. I wonder if all of these new viruses could be somehow associated with the need for so many more treatments, and like the rise of Varroa, where Varroa is becoming increasingly difficult to control. What do you think?
I think that's a great point, Landy, that, Varroa is definitely becoming increasingly hard to control. You're killing a bug on a bug. And so it is really hard. Another part of that is a lot of the viruses that we didn't have. You saw how quick the Varroa destructor virus spread across the country in 6 years. And, and is that something that, I don't know if many of you are aware of this, but the United States has their border closed to accepting bees from Canada. So you couldn't even buy Buckfast queens from Canada right now if you wanted to, because they still accept shipments from Chile. But Chile can't prove to the United States that they don't have certain viruses that we don't have yet. So there's a lot of stuff that goes on behind the scenes, but just seeing how quickly a lot of the viruses are spreading, definitely making things trickier. I also think, just as a culture, we've changed a lot in 20, 30 years. I think the number of beekeepers have sort of increased. You used to have bees at every farm just because that's what you did. You had a farm, you kept bees on it. And so the mentality's changed a little bit too, I think. So it's a lot of the factors mixed together.
Yeah, thank you. John points out the issue is not Varroa, the issue is the viruses. To control the viruses, the vector, the mite, needs to be controlled, which is very true.
Yeah. I mean,
Yeah.
some of the new research talks about, the difference with the fat bodies and stuff like that. So Varroa may still cause some problems if we had a really hard, long winter and the bees didn't have the fat bodies that they needed just from some of the Varroa damage itself. But that would be such a minuscule problem instead of almost 30 viruses,
Yeah, relative to the viruses and
Yeah.
those issues, it's like a nothing. Okay, great. You guys have more questions? John's still talking about Varroa and viruses. He says IPM does not apply to a virus. The virus is a pathogen, not a pest. I was astonished at the number of new viruses and how quickly they spread. I'd never heard of that Varroa destructor virus.
Mm-hmm.
And we don't know what the symptoms are. We just know that there's a virus and we don't know what it does.
They think it's a lot like deformed wing virus, that the bees will have deformed wing, but it's just a different virus that comes out when
Causing them.
Yes. But Lake Sinai's been around for easily a decade, and we haven't even, go Google it, guys. You can't find anything but the genome. Lake Sinai too. You can't find anything about it. So
I hope that this pandemic gets over really soon so we can get the researchers back to work and get some answers to all of this stuff. This is wild. All this new stuff is really wild. Thank you. Thank you for
you're welcome.
About it.
And
Very
I didn't
cool.
even talk about the stuff we don't have in the United States. That's a whole nother talk. But thank you for having me, guys.
Thank you very much, Megan, for doing this for us. We all appreciate it, and excellent presentation.
Thanks.
Yes, thank you, Megan. Have a good evening. Bye-bye. Good night, everybody.
Good night, everyone. Enjoy the